Уважаеми колеги, преминаваме към следващата сесия – инфекция и имунитет, първа част. Тази сесия логично е посветена на COVID-19, инфекцията, за която вече споменахме, че промени тотално нашия професионален и личен живот. В нея се очаква да чуем 3 доклада, като първия ще бъде представен от Пол Бастърд от Париж, Франция. И той е на тема Autoantibodies to type 1 interferon in patients with life-threatening COVID-19.
Добър вечер и от мен. Имам удоволствието да открия тази сесия, посветена на проблем, който наистина изпълни изцяло и личното и професионалното ни пространство, и затова сме отделили 2 части, малко повече време. Тази първа част е особено важна за нас, защото тя обединява нашите чуждестранни гости. Искаме да благодарим за тяхната отзивчивост и разчитаме много на опита, който ще споделят. С удоволствие искам да представя първият участник, доктор Пол Бастар, който е член на екипа на нашият отдавнашен познат и така сътрудник в рамките на J-проекта, професор Казанова. Docteur Bastard, c'est avec un immense plaisir que je vous introduis ce soir. Et je vous donne maintenant la parole. C'est un problème que vous allez discuter qui est très important. La différence entre dans le cours clinique qui est lié en fait différences génétiques. Donc, parole est à vous et après on va avoir la discussion. Merci.
Merci beaucoup et je suis extrêmement impressionné par votre français qui est juste parfait. Vous m'expliquerez après comment est-ce que vous parlez aussi bien. Thank you very much. So I'm going to share my screen now. Here. I hope you can see the slides. It's my great pleasure to be here with you, and thank you very much for the invitation. And so Chan and I are going to present you 2 sides of the same coin. And I will start today to talk to you about a finding we recently made, that is the finding of autoantibodies against type 1 interferons in patients suffering from life-threatening critical disease. So my name is Paul. I'm a PhD student in Jean-Laurent Casanova's lab, and I'm currently in Paris, France. And thank you very much, everyone, for attending. So the main question that the lab tries to tackle and has been trying to tackle in the last 25 years in various infectious diseases is the inter-individual variability. And so this obviously applies for COVID, and I show you here various X-ray or CT scan images of patients with very severe pneumonia, with, in the case in the far right, complete lung involvement. But the prevalence of such severe disease is actually very low. And on the other hand, most infected individuals are either completely asymptomatic or have very mild symptoms. So for COVID-19, like for the other viral and non-viral diseases, the question we try to tackle is how do we explain such inter-individual variability? So one hypothesis that the lab has been trying to study and that was applied now for COVID-19 is that in a population of people infected with, for example, SARS-CoV-2 here, some large majority will be asymptomatic or mild, and the ones that have life-threatening disease might have an inborn error of immunity that would have a genetic cause and or an autoimmune phenocopy of this disease. So to understand this, Jean-Laurent Casanova and Helen Su, a collaborator from the National Institute of Health in the United States, have led in the last 6 to 8 months a worldwide effort. This effort was aimed to answering this question of inter-individual variability, and the way that they organize this Human Genetic Effort Consortium is that we try to recruit patients all over the world. So you see here, there's more than 200 participating centers, and the samples of these patients were sent to different sequencing hubs that you see here on the left, more than 40, who would then sequence the exome or the genome of these patients with very severe disease or asymptomatic disease. So I'll let Tian in a few moments speak about genetics finding, but I will first start to talk to you about the autoimmune phenocopies of these genetic findings. So why do we know autoimmune phenocopies of inborn errors of cytokine immunity? Well, because in many cases, these autoimmune phenocopies were found shortly after or shortly before the genetic diseases themselves. So for example, for mycobacterial disease, from 1996 onwards, inborn errors of affecting the interferon gamma pathway have been shown to underlie severe mycobacterial disease, and more recently, inborn errors in this pathway have been shown to underlie tuberculosis. On the autoimmune side, autoantibodies to interferon gamma were found from 2003 onwards and were shown to also underlie mycobacterial disease. For another, candidiasis, inborn errors of IL-17 immunity have been shown to underlie CMC, mucocutaneous candidiasis, and autoantibodies were actually found in this case before the genetic inborn errors. And this is exactly the same for staph disease, and autoantibodies to IL-6 were first found actually 11 years before the first genetic inborn error of IL-6 disease was found. So what's interesting for us who are genetics lab is that these autoantibodies are in most cases genetically driven. So for patients with autoantibodies to IL-17A or F, most of them have APS1 deficiency. And for patients with autoantibodies to interferon gamma, most of them in that case have a specific HLA haplotype. What about type 1 interferons? So we studied the type 1 interferon pathway, and we've been studying it in the lab because it's been known since the 1950s that it's a very important antiviral pathway. So we know now that there's 17 type 1 interferons, 13 alphas, 1 beta, 1 epsilon, 1 kappa, and 1 omega, which is very striking because they all bind the same receptor. The receptor has two branches, IFNAR1 and IFNAR2, and Chan will soon talk about these. And upon binding to these receptors, there's a very almost immediate downstream signaling pathway that leads to the induction of downstream genes. So inborn errors, meaning genetic defects of the pathway, have been known, mostly affecting IFNAR1 or IFNAR2. And patients that lack the receptor, that are thus unable to respond to type 1 interferons, have very severe viral diseases, for example, following live vaccines or after herpes simplex encephalitis. And now we know that this gives them a higher chance of getting critical COVID-19. So what about the autoantibodies to type 1 interferons? Well, what is crazy is that they've been known since the 1980s. In patients, for example, treated with interferon alpha or interferon beta, these patients were known to have autoantibodies against the treatment. In a small minority of women with lupus, these women had autoantibodies to type 1 interferons, and we know that lupus patients have actually a high type 1 interferon signaling, and the patients with autoantibodies to type 1 interferon had lower disease activity. And these autoantibodies are also known in virtually all patients with homozygous mutations in AIR, which is a gene that causes, when deficient, a very severe autoimmune phenotype. Nevertheless, despite having these autoantibodies that were completely blocking, these patients did not seem to have severe viral diseases. This is in striking difference with what was observed in 1984 by a colleague of Jean-Laurent Casanova, Ian Gresser, who described the first patient with severe varicella. Then in 2015, our collaborator, Luigi Notarangelo, described a few patients with hypomorphic mutations in RAG1 and RAG2 who had autoantibodies to type 1 interferons, and these autoantibodies correlated with viral diseases. And finally, it wasn't until critical COVID-19 that 3 patients with APS1 were described as having critical COVID-19. So all this together led us to think and to ask the question if autoantibodies to type 1 interferon could be the cause of life-threatening COVID-19. So to tackle this question, we took the plasma or the serum from the patients that we had recruited in the COVID Human Genetic Effort Consortium, and we looked for these autoantibodies. We first searched using various techniques, and here I show you a multiplex assay for anti-interferon alpha 2 autoantibodies and anti-interferon omega autoantibodies, because these are the ones that are most known and that are used in to diagnose patients with mutations in AIR. And we found strikingly that 10% of individuals with life-threatening COVID-19 had very high titers of these autoantibodies. Our control group was actually individuals with asymptomatic infection with SARS-CoV-2. So we had a PCR diagnosis or serology, but they had not shown any symptom or even any or very mild symptoms. And none of these patients had high titers of autoantibodies against interferon alpha nor interferon omega. Next, we also wondered, what about the healthy controls, anyone in the street? Well, we found screening 1,200 individuals that only 4 of them had autoantibodies to type 1 interferons. So overall, we had identified that a substantial number of patients had autoantibodies type 1 interferons, and the next step was to confirm that these autoantibodies were functional, and so we looked for neutralization. So we've heard a lot about neutralizing assays for SARS-CoV-2, showing that the autoantibodies could block the virus, and here it's exactly the same. We just showed that the autoantibodies can block type 1 interferons. So to study this, there are several ways, and here I'm just showing 2 of them. We used, for example, healthy control cells that we incubate with healthy control plasma here on the left. And when we stimulate them with interferon alpha or interferon omega, we see a very high phosphostat-1 induction. On the other hand, when we incubate with plasma from patients with autoantibodies against either interferon alpha only or interferon alpha and omega, we see that the signal corresponding to the autoantibody is completely blocked. So here, despite interferon alpha treatment, there is no induction of phosphostat-1. And on the lower panel, to confirm that it was really autoantibody related and not due to something else in the plasma, we depleted the IgG fraction of these plasmas and showed that we could restore phosphostat-1 signaling. So phosphostat-1 is quite early in the signaling pathway. So we also looked more downstream at the interferon stimulated genes. And here I just show you one of the antiviral genes that we looked at. And we see that at 2 hours after stimulation, there's a complete blockade of interferon alpha 2 induction of CXCL10, for example. And interestingly, you also see that 2 individuals here had neutralizing autoantibodies to interferon beta. So this leads me to ask the question, what about, we've looked at 2 cytokines, interferon alpha 2 and interferon omega, but what about the 15 others? So we did several assays to assess this, and here you can see the ELISA results for the 17 type 1 interferons, and which shows that most patients actually recognize the 13 alphas, which makes a lot of sense because on the right side of the slide, you see the phylogenetic tree, and you see that the 13 interferon alphas are almost identical. And most patients recognize also interferon omega, which again makes sense because looking at the phylogenetic tree, which shows the structure homology, we see that interferon omega is closely related to interferon alpha. And on the other hand, very few patients had autoantibodies to interferon beta, kappa, and epsilon. And this is makes sense because these interferons actually more different than the alpha and the omegas. So we've shown that most patients have autoantibodies to 13 alphas and to interferon omega. But what happens in vivo? So to assess the in vivo effect of these autoantibodies, we measured the levels of circulating interferon alpha. Our hypothesis here was that if these autoantibodies were also neutralizing in vivo, they could perhaps lead to an abolished circulating interferon alpha. And so that's what we see here. When we measure circulating levels of interferon alpha in patients with autoantibodies, we actually don't detect circulating interferon alpha or at very low levels, whereas when we measure it in patients with COVID and without autoantibodies, we see much higher levels of interferon alpha in the blood. And finally, to assess the effect of these autoantibodies against the replication of SARS-CoV-2, we teamed up with the group in Charlie Rice's lab, who recently got the Nobel Prize, and they performed a very elegant experiment. So what they did here was to take control cells, infect them with SARS-CoV-2, treat them with interferon alpha, and then, well, at the same time, actually, add plasma either of healthy control or plasma of patients with autoantibodies. And the idea here is that if the plasma of patients with autoantibodies was able to block interferon alpha, then it would lead to higher viral replication. And so that's exactly what we saw, which is that when we look at the blue line here, which are actually the two healthy controls that we used, well, they don't block the antiviral effect of interferon alpha, and there is no viral replication. So this shows that healthy control plasma does not impede interferon alpha's antiviral effect. On the other hand, all the gray lines here are patients with life-threatening COVID-19 and autoantibodies, and we see that in all of them, actually, when we incubate plasma of these patients with interferon alpha, it leads to very high SARS-CoV-2 replication. And this is the case to very low dilution. So here in the lower part, you see the dilutions that we used. And when we dilute up to 10 to the minus 5 the plasma, we still see that the plasma of these patients is able to block interferon. And finally, this is a very potent effect also because when we incubate with a monoclonal antibody, well, we see the exact same effect. So that means that the autoantibodies in our patients have actually a very high affinity to interferon alpha 2. So this is over with the experimental data, and I'll just show you quickly an interesting observation that was made in the patients with autoantibodies. So regarding ethnicity, we looked by PCA analysis on genetic data at the ethnic origin of our patients with autoantibodies, but I actually found no significant difference between the patients with autoantibodies in red and those in black without autoantibodies. On the other hand, what was very surprising is that 95% of the patients with autoantibodies were males. And furthermore, about half of the patients with autoantibodies were above 65 years old. So this reminds us of the two main risk factors of having a severe course of COVID-19, which is to be a male and to be old. So the presence of these autoantibodies could potentially be a first explanation of why older patients and why male patients have more severe disease. So overall, we found that 10% of patients with life-threatening COVID-19 have autoantibodies to type 1 interferons. And I might mention here that we think that these autoantibodies were pre-existing to the disease for several reasons, but including two important ones, which are that we found 2 patients for which we had samples from before COVID, and the autoantibodies were already present before COVID-19. And we also realized that these autoantibodies were IgGs, and given the fact that the samples were drawn very early during hospitalization, it's unlikely that these patients had a very high IgG response so early. So it's most likely that in all these patients, the autoantibodies were present before infection. and are thus causal of the disease. We also showed that these autoantibodies could neutralize type 1 interferons, both in vitro in our assays, in vivo with a low interferon alpha level, and they block the protective effect of interferon alphas and the 17 interferons most likely against SARS-CoV-2. And finally, they're mostly present in males, and half of these individuals with autoantibodies are over 65. So our model would be that patients, that 10% of patients with life-threatening COVID-19 have autoantibodies pre-existing, here in red, that block type 1 interferon signaling once these individuals are induced with the virus, and this leads to an abolished antiviral response and a high viral replication. So this has important implications. For example, we should screen for these autoantibodies in patients with COVID-19. It would also be important to screen for autoantibodies against type 1 interferons in patients that are donors of convalescent plasma, because we wouldn't want to transfer these autoantibodies to another patient. And also we could think of removing these autoantibodies or using interferon beta treatments early in the disease, because most patients did not recognize interferon beta. So with that, I'd like to, thank you very much for the invitation, and thank all the physicians, the patients, the families that have made this study possible, the whole lab, both Paris and New York. And as you can see here, many co-authors, first from the NIH, Lindsay, Helen and Luigi Notarangelo, who've been very helpful in these experiments, Chian, who's been crucial for the whole project, Charlie Rice's team that have hugely helped with the viral experiments, and of course, Jean-Laurent and Laurent Abel, who had the lab both in Paris and New York. And I'm happy to take any questions now. I don't know if it's better now or after Chian's talk.
Thank you very much. So we have now a few minutes for questions. And
I'd like just ask a very speculative question. Thank you so much, Dr. Buster, for this interesting data. Can you speculate whether we can neutralize these autoantibodies against type 1 interferon using any kind of biological therapy, namely monoclonal antibodies against these autoantibodies, such as anti-idiotypic approach? Do you think it will be an encouraging approach?
I think it would be great to have them. I don't know of any ongoing trial or ongoing treatment for this, but I think that given the percentage of patients with autoantibodies, the companies and researchers working on that would definitely be interesting. We can also think of removing these autoantibodies with plasmapheresis, or hopefully specific plasmapheresis to remove specifically the anti-interferon autoantibodies. It would be great, yeah.
Thank you.
I have a question also. It's really seems a serious problem we haven't much thought about previously when treating patients with reconvalescent plasma. But how do you estimate the frequency of these autoantibodies among the general population? And did you have any idea if these patients with life-threatening COVID have had any other severe viral infections in their past, or you didn't have the chance to get this data?
Yeah, that's a very important question also. So it was hard to get for the patients in this study, because what I didn't say is that 40% of them, and probably more, were unfortunately deceased because of their COVID infection. And for the others, we didn't know of any major viral infection previously. But we are now screening the other viral cohorts that we have in the lab, and actually find that patients with autoantibodies can also have other viral diseases. So I think that if we were to ask these patients with autoantibodies if they had other viral infections, it's most likely that some of them at least would say yes.
Yes, thank you. So if there are no more questions,